Hi guys, welcome to my YouTube channel. In this video, I will discuss about electron transport chain. So let's start. In this session, I will cover electron transport chain definition, electron transport chain location, electron transport chain components and its carriers, electron transport chain equations and the chain's complexes, electron transport chain steps, and electron transport chain products. So at the first, you have to know what is electron transport chain. The electron transport chain is a cluster of proteins that transfer electrons through a membrane within mitochondria to form a gradient of protons that drives the creation of the adenosine triphosphate. ATP is used by the cell as the energy for metabolic processes for cellular functions. Here you can see the whole processes, and here some complexes are also involved in these processes, but I think you see this process very complicated, but I assure you I will make this process very easy or simple to you. The location of the electron transport chain. During the processes, a proton gradient is created when the protons are pumped from the mitochondrial matrix into the intermembrane space of the cell, which also helps in driving ATP production. Often, the use of a proton gradient is referred to as the chemiosmotic mechanism that drives ATP synthesis, since it relies on a higher concentration of the protons to generate proton motive force. The amount of ATP created is directly proportional to the number of the protons that are pumped across the inner mitochondrial membrane. Okay. So electron transport chain, there are some carriers. One is the FMN, that is flavin mononucleotide, and other is ubiquinone, that is also known as coenzyme Q, and the another is the cytochromes. Okay, the electron transport chain equation. So here's the equation. The electron transport chain consists of a series of oxidation reduction reactions that lead to the release of energy. A summary of the reactions in the electron transport chain is, that is NADH plus half molecule of O2, and then H plus, then ATP plus inorganic phosphate that make NAD plus ATP and H2. Okay. So you can see in this processes there are several complexes are also involved. This is complex 1 and complex 2, complex 3, complex 4, and this is complex 5, or named as ATP synthase. It makes the ATP by the uh, use of protons. Okay, so now I will talk the complexes, each name of the complexes. The first complex, that is the name of the first complex is NADH dehydrogenase. NADH dehydrogenase. And complex 2, that is known as the succinate dehydrogenase. Okay, so it contains iron and succinate, and which oxidizes FAD to the FADH. You know that. And coenzyme Q that accept electrons from the FMNH2, and that it is the complex one. It contains some, it's accept electron from the FMNH2. So okay, so this is the coenzyme Q. Here is present the coenzyme Q, and this is the complex one, that is also known as NAD dehydrogenase. Complex 2, that's known as the succinate dehydrogenase. And complex 3, is also known as cytochrome B. And complex 4, that also known as cytochrome A. Okay, this is a complex 4 and known as cytochrome A. So cytochrome A, that it contains a heme group, in which the Fe3 plus accept electron from the cytochrome C to become Fe2 plus and transfer electron to the O2. Okay, I will discuss it later. And complex this 5, and it's also known as ATP synthase. Okay, next. So this is the whole processes, and here you can see this is in the matrix. You can see that a charge is negative, and as there is low H plus or protons, there is low, and here is a high concentration of the protons in intermembrane space, as the proton pumped in the intermembrane space from the matrix by this using of complexes. Okay, here is the main features of all the is very detailing image of this process. So let's discuss one by one. Start from the complex one. So the point is the transfer of electrons from NADH to coenzyme Q. Uh, so the first complex is NADH dehydrogenase. NADH dehydrogenase, and another name of the another name is here, that is NADH coenzyme Q oxidoreductase. You know that. So at first, you have to know how the NADH get produced. So that the NADH, that it is produced by alpha ketoglutarate dehydrogenase, isocitrate dehydrogenase, and malate dehydrogenase reactions of the TCA cycle. Okay, by the pyruvate dehydrogenase reactions, and that converts pyruvate to the acetyl CoA by beta oxidation of the fatty acids and others oxidation reactions. You know that. So here this is, the NADH, how I say that how the NADH is produced. That processes alpha ketoglutarate dehydrogenase complex, isocitrate dehydrogenase, and malate dehydrogenase reactions of the TCA cycles. Okay, so here NADH that oxidize, and it donate this sugar byproduct, donate 2 electron to the FMN. Okay, so FN, the name of FMN. This is full name is flavin mononucleotide, and it donates electron to the FES. It's iron and sulfur. Okay, and iron and sulfur. This there is a series of FES, and what their function? FES collect the energy of the electron and help the complex one to form the proton from the matrix to the intermembrane space, and it pump 4 proton. So, and another function is FES also collect this electron and serve to the and give to the coenzyme Q. Okay, and here is another. It is the function of complex one, and what the function? It transfers the electron to the coenzyme Q, and another is it pumps 4 protons from the uh, matrix to the intermembrane space. Okay, so the next is the complex 2. Complex 2, that is also known as succinate dehydrogenase. Succinate dehydrogenase is the complex 2, and the main difference from the complex 1, that succinate dehydrogenase doesn't involve in the proton pumping. So it doesn't pump proton like the complex 1. Okay, so the electron from the FADH2 produced by the reaction, such as the oxidation of the succinate to the fumarate, and enters the electron transport chain at the complex 2. The switch contains succinate dehydrogenase, and complex 2 will transfer the electron to the coenzyme Q without associated proton pumping across inner mitochondrial membrane. So I will repeat that electron from the FADH2, that is a byproduct of sugar, that produced by reactions such as oxidation of the succinate to fumarate, entered the electron transport chain at complex 2. So at complex 2, which contains succinate dehydrogenase, and complex 2 will transfer electron to the coenzyme Q without the associated proton pumping across the inner mitochondrial membrane. It, and then I will discuss that the transfer of the electrons. How the transfer of the electron from the coenzyme Q to the cytochrome C. The complex 1 and complex 2 both donate electron to the coenzyme Q. Okay, and now we will see that coenzyme Q transfer the electron to the cytochrome. Coenzyme Q, how it donate the electron to the cytochrome. Let's see. And here coenzyme Q that passes electron through FES centers, that that in complex 3, that is also known as cytochrome BC1 complex, cytochrome BC1 complex, or cytochrome B. So this it has several components of complex tree. And the coenzyme q that passes the electron coenzyme q passes the electrons to FES centers. Here there's some FES center, and coenzyme q passes the electron to the FES centers. Okay. And and the centers to the cytochrome B and C1. And cytochrome has a B and C1. It also involved here. And cytochrome Q passes the electron to the FES centers to the cytochrome B and C1, which transfer the electron to the cytochrome C1. And so FES transfer the electron to cytochrome C1, and cytochrome C1 transfer the electron to the cytochrome C. Clear? So again I repeat. So enzyme Q donate the electron to the FES, and FES donate the electron to cytochrome C1, and cytochrome C1 donate the electron to the cytochrome C. Okay. So here again also one is recycled again with the cytochrome B. So please remember that uh, it is the protein complex involved uh, in the transfer is called complex 3, or the cytochrome BC1 complex, and this complex is also known as coenzyme Q-C1 oxidoreductase. Thus, the cytochromes in each contain heme. The cytochromes each contains heme as a prosthetic group, but have different proteins. Okay, and here the cytochrome can carry only one electrons. It only carry only one electrons, and one is recycled again, and then it will go to the cytochrome. Okay, so because the cytochrome can only carry one electron at a time, and two molecules in each cytochrome complex must be reduced for every molecule of NADH that is oxidized. In this ferric Fe3 states, the heme ion can accept one electron and be reduced to the ferrous. You know that Fe3 plus to the Fe2 plus, and then it will vice versa. It make vice versa and come to its own previous state, Fe3 plus. So here this is the complex 4. This is the complex 4. Now we will see how cytochrome donate the electron to the oxygen. Let's see. This is the very interesting processes, the transfer of the electron from cytochrome C to the oxygen. Here, that this complex 4, it also contains cytochrome A, cytochrome A3. Okay, this complexes. So, cytochrome C transfer electron to the cytochrome A3. Cytochrome A, cytochrome A3. It transfers the electron cytochrome A. This. Okay, and this also contain this A3 also contain a heme group and two different proteins that each contain copper. So this part cytochrome A and cytochrome A3 that also contain copper. The type of copper that's here that copper is CuA and copper B. So it contains two copper. So that the cytochrome A and A3 each contain a heme and two different proteins that each contain copper, and different proteins that are called CuB and CuA. Okay, so two electrons are required to reduce one atoms of oxygen, and therefore each NADH that is oxidized, one half of the O2 is converted to the H2O. It's very interesting. So in this process, there are two proton, two electron, and half molecule of oxygen is needed to make one molecule of H2O. So half O2, two H plus, and two electron is needed to make this H2O. And two electrons, power of two electrons, it's needed to pump the proton. Here that the number of protons is very less, as the two electron is used in this process, and two electrons is used to pump the proton here. And in this that in the tree that they also use this energy from the electron and pump proton from in matrix to the intermembrane space. So it was the transfer of the electrons, and here is the ending. That here it makes H2O, and now I will talk about the uh, here the the energy generation or ATP, how the generation of the ATP in etc. So in this process, you see that the complex one also pump 4 proton, complex 2 doesn't pump any proton, and complex 3 pump 4 proton, and complex 4 pump 2 proton. So total number of protons are 10. 10 protons are pumped in the intermembrane space from the mitochondrial matrix, and and the production of the ATP is the production of the ATP that is synthesized from the ATP synthase here. And you know that ATP synthase also used 4 proton to make 1 ATP. 4 proton to make 1 ATP. So from the NADH that pumps 4 protons from the complex 1, and from the NAD is this electrons using this energy or electrons, you can see the 1 NADH that will make 2.5 moles of ATP. How I calculate this? You divide 10 by 4. As you know, the 4 protons make 1 ATP. So 10 protons. So here the one ATP that makes here you can easily calculate the exact amount of the ATP that is generated by this process and has not been clearly established. Okay, so it has not been clearly established, but current thought indicates for each pair of electrons that enters the chain from the NADH, 10 protons are pumped out of the mitochondria. That's clear. As it takes 4 protons to flow through ATPase to synthesize 2.5 moles. As I divide 10 by 4, so you know that 2.5 moles of ATP is that has been generated from the 1 mole of NADH. Okay, same as for every mole of FADH2, you got 1.5 moles of ATP. Okay, so it was the energy calculation, and now we will talk about the significance of the electron transport chain. So it has an important role in both it has been important it has an important role in both photosynthesis and cellular respiration. And you know that from the glycolysis and cytic acid cycle, they make necessary precursors, the electron transport chain that makes the energy from the electron transport chain, and the majority of the ATP that is created from here. And the electron transport chain is the final and the most important step of the cellular respiration. Okay, so it was the significance or importance of the electron transport chain. So it was here I end the video, and now finally I will talk about the total ATP. From the glycolysis, you get directly to ATP, and 2 NADs. One NADs makes 2.5 moles of ATP, so 2 NADs will make in eukaryotic cells 5 moles of ATP. Here, the pyruvate oxidation you get directly in pyruvate oxidation you get 2 NADs that makes 5 ATP again, and citric acid cycle that makes directly 2 ATP and 6 NADs and 2 FADs. 6 NADs it means 15 ATP, and 2 FADs it means 3 moles of ATP. So totally you get 30 to 32 ATP in number. Okay. So this session has been ended here, and thanks for watching. And please like the video. If you like this video, please like and share with your friends, and subscribe to be a member of our channel, and hit the bell icon to get such kind of video. Thank you.